This is my home hospital, Cedars-Sinai Medical Center in Los Angeles, and we have about 1,100 beds in Cedars-Sinai and Center for Regenerative Medicine. But I'm from Department of Neurosurgery, and actually my research is focused on translating space technology from NASA. I have a dual appointment with NASA into neuroscience, and I will explain to you several of those clinical trials and how that could actually help stem cell research. But also I want to talk about a new emerging field of nanobioelectronic, which is basically integration of nanotechnology with device and imaging and basically cellular therapeutics or stem cell. As our colleague mentioned, I was heavily involved in President Obama's BRAIN initiative, and now we are involved in the G20 World Brain Mapping Initiative, and I will talk about that. I'll give you an example of nanobioelectronic technologies, and I have like two short videos to show you and 28 slides. So here we go. I would like to thank my colleagues who have been collaborating with me from NASA and also from the White House Office of Science Technology Policy, as well as U.S. Department of Defense and Cedars-Sinai Medical Center. Full disclosure, shareholder and patent holder in a Microsurgeon is a device company that is a spin-off out of an invention that I had to microwave brain cancer, so you'll get to hear about that. And in terms of the objectives of the talk, I'm going to talk about the history of and the role of Society for Brain Mapping and Therapeutics in translational neuroscience and President Obama's BRAIN initiative. Define brain mapping. What is brain mapping? What is nanotechnology? Nanoneuroscience, nanoneurosurgery, and nanobioelectronics, and provide examples of those technologies, and also in the context of brain cancer and neurotrauma. Then I will talk about a little bit about SBMT's G20 Brain Initiative, and provide an overview about the G20 Brain Initiative. So a little bit of history. We started the Society for Brain Mapping and Therapeutics about 2003 when the Facebook started in collaboration with NASA and Caltech and USC, where I was doing my fellowship. And the mission of the organization is improve the diagnostic, treatment, and rehabilitation of patients afflicted with neurological disorders. The approach is basically multidisciplinary. We bring scientists, engineers from different disciplines that they usually don't meet in annual meetings and publications in consortia to address neurological disorders. So the vision of the organization, as I mentioned, is basically building a worldwide collaborative network to address the neurological disorders. Our goal is to apply gap research to improve understanding, basically education, care, and public policies which affect management of neurological disorders. So this is a picture. We actually annually award a scientist who has shed light on neurological disorder. This is Stephen Hawking, that we presented him with an award because of his role in shedding light on ALS. So we have different programs, annual meetings and symposias and advanced training programs, fellowship visiting scholar program, and then student chapters, industry partnership, and publications. And we are currently starting to partner with IEEE and SPIE, basically translating many technologies into neuroscience. These are annual conventions, started from 2004, and comes to our publications. It's basically 3 journals in neuroimage. We have our own journal with PLOS ONE, Neuromapping and Therapeutics, and this is a textbook of nanoneurosurgery that I'm the editor. We published it, and President Obama received a copy, and I got a letter from him. So in terms of public policy, how do we actually interact with the U.S. Congress? If you look at from this side, with the congressional leaders, we actually educate them in a brain mapping day at the U.S. Congress. And this is a congressional language that we wrote, and basically with the head of the NIH, we pushed the policy for Brain Initiative, and this is President Obama who introduced Brain Initiative. And also we get Hollywood stars involved in terms of basically educating public about neurological disorders. This is Dustin Hoffman receiving an award for his role in raising awareness about autism. So, in the Brain Initiative, we had 3 recommendations for President Obama. Formation of national data repository and analysis for neuroscience, creation of national network of brain procurement, allocation, and banking, and establishment of National Alliance for Nanobioelectronics. So the first one is basically focused on creating a database in terms of who is doing what. Basically, instead of duplicating efforts, we basically have a database and start collaborating together. Second part of the initiative is basically creating a network of brain banks. So we actually provide tissues to scientists who could actually investigate human brain. And the third one is basically integration of nanotechnology, stem cell, and device and imaging. So basically, what is the definition of brain mapping? So brain mapping, imagine like a Google map. You get a snapshot of the Earth, that's your MRI. You zoom it down to a city, that's the brain structure. Zoom it down, see the basically highways, that's brain connectomics. You zoom down further to a local area, so those are particular regions of the brain, and then you see the cars, those are cells, people in the cars, those are genomic particles. So basically, brain mapping from the Brain Initiative standpoint involves from the imaging all the way down to the nanoscale. And application of nanotechnology in neuroscience is basically nanoneuroscience, and the therapeutic application of nanotechnology, which could enhance neurological treatment, called nanoneurosurgery. So, and the combination of stem cell or cellular therapeutics with nanotechnology, device imaging, and mathematical modeling basically is called nanobioelectronics. So this is one example of nanobioelectronics. So we took actually NASA's carbon nanotube that they use in shuttle, and we functionalize it. So you have basically, this doesn't fall on me, if you have carbons, you basically functionalize these carbon-carbon connections, and carbon connection can be actually degraded. So I have a video to show you. So, and if I can bring the video here. Okay, so this is actually live. So this is live macrophages under confocal microscopy. The red dots are actually NASA's carbon nanotube that are functionalized, and they're picked up by these macrophages. So you could see them actually moving around. This is to activate the cells so they can actually attack brain tumors. If I can bring it up again so you could basically see it. So same type of nano platforms could be used to deliver drugs or be used as a scaffolding structure for actually stem cells. So this is not running right now, but oh, here we go. So again, for you to see, these are carbon nanotubes. So we use them to activate macrophages to attack brain cancer. All right. So we wanted to actually look at we want to see if we actually use these carbon nanotubes for delivering siRNA or DNA to, this siRNA or DNA to cells. And they come across an interesting finding that the ratio of loading of one carbon nanotube to one siRNA was 1 to 1. So that's very good in our dose analysis. And also in the carbon nanotubes, we realized that some of the cytokine profiles that actually change a little bit. Here is IL-10, tumor necrosis factor alpha, and IL-1, but they actually stabilize. So the cells are actually tolerating this. Although the systemic injection of these carbon nanotubes are really not recommended, and we come up with another nano platform that I will go over with you. So this is basically the blue areas are brain tumor. This is in vivo of the same technology. We injected the carbon nanotubes here, and these are carbon nanotubes spread out through the tumor area, and the green structures are macrophages. So you can see some of the macrophages actually taking these carbon nanotubes, and that could actually help for kind of immunotherapy of brain cancer. So one of the problems with carbon nanotubes is that actually they activate the complement system. So it's not a good idea to inject the carbon nanotube to the bloodstream or inhale it. So because of that, we come up with another nano platform, which is built in our lab at Cedars-Sinai. Is a nano prodrug that the structure is very simple. Is ibuprofen. There are 2 molecules of ibuprofen and tagged with basically tetra ethylene glycol, which is basically is kind of a spacer. So you have spacer in between, so you can load different molecules. Depends on what you want to treat. In this case, we want to treat brain trauma. So we use ibuprofen and alpha ketoprol, and basically did a spontaneous emulsification, and basically finally we tag this with a fluorescent dye. So you could actually, in animal model of brain trauma, you could actually see where the distribution of nanoparticles are, and it basically you can pick up the signal. This nanoparticle could be used actually in stroke. As I mentioned to you, we use these spacers, and basically in between spacers you can deliver different drugs, and you can use it in Alzheimer and chemotherapy and neurotrauma. So one of the work that I did when I was at City of Hope, we wanted to actually visualize where these stem cells actually go throughout the brain tumor, and we wanted to know if we could really come up with a novel kind of method of visualization for this. So this is a paper that we published there, and I'm not going to go through the details of it, but just we did this animal study and use confocal microscopy to kind of estimate the primary tumor, the boundaries of primary tumor, and then see where the kind of stem cells go after injection. And it was an interesting finding that the stem cell distribution was about 70 to 90% throughout the tumor, basically microenvironment. So this brings me to the importance of using predictive modeling and mathematical modeling in studying of stem cell or cellular therapeutics in general, and importance of the technology, which basically this is another technology we use to from NASA to sniff brain cancer and skin cancer. It turns out that their readings are different, and they're totally could be differentiated. And right now at Cedars-Sinai, we are actually trying to kind of get the different stem cell lines and test them and see if we could pick up different signals in terms of smelling the cells. In terms of physics of waves, again, going back to concept of nanobioelectronic, I'm giving you some examples in terms of brain tumor therapy. In terms of physics of waves, we are focusing on UV imaging with NASA in the spectrum of 10 to 400 nanometers, and also infrared, 700 nanometer to 1 millimeter, and microwave, which I will discuss that. So for intraoperative brain mapping, we are actually using NASA's infrared thermography to delineate the margin of tumors. This is a melanoma, metastatic melanoma to the brain, and you can actually get the margin of the tumor. And now we are actually testing generation 2 of NASA's ultraviolet imaging in the operating room, which could help us somewhat delineate margin of brain tumors, like, no, not this one. Actually, it's the wrong one. This one probably. Yeah, so in this video, you will see that it's very difficult to delineate the margin of glioblastoma multiforme. But through ultraviolet imaging, we may be able to actually see where the tumor is. So that's the benefit of using kind of NASA's technology. So I'm going to just move very quickly, so we are a little bit behind. And that brings me to our device that we invented. Basically is a microwave ablation device that Dr. Keith Black and I invented at Cedars-Sinai and got an FDA approval. You can see that we can actually use this device to make a lesion in the brain. This is a brain of the pig. And the device looks like this. Very simple, works with any computer. And you can make a lesion in the brain and basically get away from a major craniotomy procedure to a pure-hole surgery. But also it's interesting that you can heat up the brain to a certain level to open up blood-brain barrier and basically inject nanodrugs to basically attack brain tumors. So that's again bring me to the importance of using cellular therapeutics with device imaging all at the same time, which is kind of called a nanobioelectronic approach. Based on this type of science, we last year at the G20, I was on the panel of the Innovation 20 when President Obama was in Australia. So we discussed importance of using technology to reduce cost of care. As my colleague, Professor Maria mentioned, the cost matters. And because of that, I think we have to come together as scientists, surgeons, and engineers to form consortium and collaborative effort to reduce the cost. So we published our first initiative paper last year, and the focus of the initiative is now really building a global alliance in nanobioelectronic and brain mapping and therapeutics. And as part of the G20 effort, we would like to build a consortium of scientists, as I mentioned, and basically coordinate a global response to rising costs of neurological disorders and global harmonization of the related policies and standardization of data. In terms of standardization, we are collaborating with IEEE now, because most of the technologies are used in brain mapping are actually designed by engineers. And in terms of facilitating translation of technologies across disciplines of science, we would like to have series of interaction with the parliaments of Canada in this case, but also G20 to translate this technology to clinical practice. And identifying global regulations and guidelines on clinical trials that could help translation of such technologies, and also global partnerships. So by now you know the history of the Brain Mapping Foundation and Society for Brain Mapping and Therapeutics and its role in translational neuroscience. You know the definition of brain mapping, basically from imaging all the way down to nanoscale. And I provided you with a series of examples on nanobioelectronic examples, nanoneurosurgery, and nanoneuroscience. And you're aware that the President Obama's Brain Initiative is actually focused on advanced technologies that could actually reduce costs. And we are welcoming you to be part of the G20 Brain Initiative. So in conclusion, we know that nanoneuroscience, nanoneurosurgery reduce basically impact the field heavily in the future. And the only way to advance therapeutics is through partnership. And integration of stem cell with nanotechnology device and imaging. And we think that scientists should be really involved in global policy in neuroscience. And such partnership requires sound policies. And basically, Brain Initiative could provide a platform in this area. And also, we welcome our colleagues here in this audience to be part of the G20 Brain Initiative. And thank you very much.